Our next speaker, I totally forgot to print off her introduction, so she'll wing it here. She'll do it for us. Our next speaker is Dr. Jackie Kraveka. She's going to be doing a discussion on DFMO updates and BCC trials and the BCC research. So we need her slides.
Hi, everyone. Well, the slides are coming up. I'm just honored and so happy to be here with you all. I'm a pediatric oncologist at the Medical University of South Carolina in Charleston, South Carolina. I'm the PI for our COG studies and have been very involved with the Beat Childhood Cancer Research Consortium from the very beginning and have been involved in development of trials and serve on its executive committee. And how do I advance? Right. I think that's working. These are my disclosures. And some of you may have heard about BCC. Originally were founded by Giselle Scholler in 2006. I was speaking on her behalf. Her daughter's graduating from college today, so I'm happy to fill in. But BCC started, it was called NMTRC, or the Neuroblastoma, Medulloblastoma Translational Research Consortium, and then started in 2006 and has grown to over 50 universities and hospitals. It really focused on partnering with pediatric research scientists and take what's kind of being studied in the lab and take it over into clinical trials. And our goal is to find a cure for all children with cancer. This is just some of our leadership in BCC with Dr. Scholler and Genevieve and Abby, who are our program managers, and our bioinformatician and biostatistician, as well as the research lab. These shows are 50 plus sites in the US and Canada, and we are expanding to Spain with Dr. Moore in Barcelona and Dr. Ambar in Brazil as well, and that's in process. I'm going to go talk about a little bit of our current open studies. These are the studies we currently have available right now, and touch briefly on it. But, kind of walk you through and talk a little bit about DFMO or fluoronethine, which many of you are very, very quite familiar with. So we know that relapse is one of the major causes of mortality for our kids with high-risk neuroblastoma. And relapse occurs really about 30% of patients within 2 years. You really have to do more to prevent relapse. And our studies on DFMO really focused on relapse prevention. And the reason is that once a patient relapses, survival is really poor. So really need to help prevent relapse so that our children can live long-term lives. So a little bit about DFMO or fluoronethine. I know we've touched on it today. So there's an enzyme that's called ODC, or ornithine decarboxylase. And it's the rate-limiting enzyme in these proteins called polyamines. And polyamines are involved in cell growth and cell proliferation. So DFMO does, it blocks ODC, and then that decreases polyamines. And then what happens when you do that, it leads to prevention of cell growth. There's what's called this Lin28, Let7 pathway that kind of is a pathway for cancer stem cells. So we really think how DFMO works is really cytostatic. It's not going to kill the tumor cell, but keep it from growing and keep it from relapsing. And that's how our mechanism of action is really targeting that cancer stem cell. Oops, I think I turned it off. There we go. So how we started was a phase 1 trial, which was a dose-finding study for neuroblastoma. And what we found is that DFMO was safe at all dose levels. We didn't reach any dose-limiting toxicity. And what was done, 3 patients were long-term survivors who went on this study with DFMO and also some oral low-dose etoposide. So that really led us to go and found and start a phase 2 trial. And we know one of the exciting things really that last decade was the work from Dr. Yu is with anti-GD2 antibody, dinutuximab, that really improved survival and became the standard of care. But unfortunately, a lot of kids still relapse. So we thought, we need to do something better to prevent that. So what we did is that it was a phase 2, it was not randomized, it was a single arm study across BCC sites, and DFMO was given to children who were in remission, either at the upfront therapy or kids who are relapsed or refractory. So I'm going to talk about really that first group of patients, those kids who were in remission after upfront therapy, kind of following the standard therapy, per COG. And what we saw is that we really saw an increase in both event-free survival and overall survival. But one of their critiques was this was not a randomized study. This was a single-arm study. So we really, sought to do a comparison. And COG was able to provide us the data from Dr. Yu's study and BL032. And we were able, with the statisticians, to really look at historical control and something that's called propensity score matching, which I won't get into. I'm not a statistician, I know. And basically, using propensity score matching, or PSM, it's a mouthful, we match patients on these 11 variables that are shown here. And that, based on that, we were able to really do a comparison for the kids who were treated identically, who received Dfmo, and those who did not. And what that PSM showed, it confirmed that compared to that historical control, one thing that this was an older study. These were patients, the majority of which received a single transplant, not tandem, which is now our standard. There was an increase in event-free survival, and overall survival. We do have a poster on the 7-year survival. That's I think, as a rapid fire session. And one of the things that we can ask, well, what are the side effects of DFMO? And this is data from 2 of our studies looking at safety. So the main thing is about 11% of patients do have some hearing loss, most of which is reversible and are dose reductions built in. But overall, it's a drug that at the dose of 750 milligrams per meter squared twice daily is overall, very well tolerated. And we were excited that it was approved in December of 2023. And it's called iWilfin, and this is Will and Finn. Will was one of the patients who was on that phase 1 study. And his father, Pat Lacey, helped fund a lot of the trials. And then Finn is the nephew of one of the vice presidents of US World Meds. And that's why it's called iWilfin. And it is now commercially available. And we're US World Meds is, working on getting regulatory approval in Europe, also in getting it available in China as well. There are some compassionate access programs available in other countries that do not have access to it. I'll go and talk about our other DFMO trials that we still have open. 06 is open at Penn State, and it's an expanded access program really for patients who would not qualify for commercial product, perhaps those patients who have overexpression of Lin28B and other tumors. And I'll go briefly about our other trials as well, if I know time is limited. NMTRC14 was a maintenance trial, and most of the arms are now closed. And one of the things that we were doing, these were patients who were in remission up front following standard therapy. We're comparing 2 dose levels because some patients still relapse. And the only arm that's open is at that higher dose of DFMO, and that's almost met its accrual goal. And then NMTRC012 was a trial that we looked at adding a targeted agent to upfront therapy. During induction, patients went on to transplant and then were randomized to receive DFMO either with concurrent immunotherapy, and all patients received DFMO as maintenance. And these were the targeted agents based on all patients were sequenced and based either on a DNA mutation or RNA overexpression. They went on to receive one of 4 targeted agents. Part A, that induction arm, is closed, and we don't have results yet, but Part B is still open. So Part B is that immunotherapy arm, which all patients receive DFMO at the end of immunotherapy, but half are randomized by a computer to receive DFMO either at the beginning or the end. And that's an ongoing study at our sites. And then 015 is another study. It's a little bit older. That's almost met accrual as well. That's probably going to close in the next couple months. And the arms that are open are for patients with relapse and refractory disease at a little bit of higher dose of DFMO of 1,000 milligrams per meter squared. And they receive DFMO concurrently with oral etoposide for 6 cycles and then continue maintenance Dfmo for 2 years. That study is also almost met its accrual goal. And then there's also a session on Dfmo with Dr. Hogarty, Dr. Scholler, Dr. Cohn. I believe it's on Monday morning at A&R for to I think if you have any interest. So let's talk about our current open studies now. BCC 018 is an induction study, and we're looking at adding naxitamab as an anti-GD2 agent to upfront therapy. So you've heard a lot about GD2. So GD2 is a glycosphingolipid. It's a fat with a sugar on it, and it's expressed in all neuroblastoma cells. And there are several antibodies being developed. And really, Dr. Yu's study, NBL 032, really showed a big difference in adding immunotherapy post-transplant post-consolidation, improving both event-free and overall survival. Similar effects were seen using dinutuximab in Europe. And then, as people have mentioned, the use of chemoimmunotherapy for patients with relapsed and refractory disease has really been exciting for all of us to really help our patients. And one of the things, like how this idea came about, was based on the work of Dr. Firm and Dr. Federico from St. Jude's, is that St. Jude's study, NB 2012, and I believe there's a follow-up that's an oral presentation at A&R, they added their antibody to the COG backbone and really saw impressive end of induction responses, which are almost like 68% versus 43%. And our thought is if you have a better response at the end of induction, that's going to correlate, much better with outcome. So those results were really exciting. That's kind of what kind of led us to do this trial in COG, is taking the similar approach with dinutuximab. So naxitamab is one of the other antibodies that's available commercially in the U.S. It was a humanized 3F8 antibody that was developed at Memorial Sloan Kettering and licensed to Y-mAbs. It was approved in November of 2020, and its current approval is for patients that are more than a year of age with relapse or refractory disease who have disease in the bone or bone marrow. This is what our schema looks like. We're adding it to the standard backbone for neuroblastoma in the U.S., so patients will receive naxitamab with every cycle. And then our overall view is that this study is an induction-only study, so patients go on and receive their induction, and then they go on to consolidation with stem cell transplant, and then go on to maintenance therapy and can then enroll on the 012 study as well. We did built in some cycles for salvage for those patients who are poor responders. And we actually have an amendment right now that we initially had made that last cycle to be cycle containing doxorubicin, but really want to avoid cardiotoxicity in our patients. So we're going, and patients can receive up to 6 cycles of chemoimmunotherapy with naxitamab, or temozolomide, which is called the HITS regimen, and patients get evaluated every 2 cycles. We have another study that's just opened at the University of Florida and Penn State. Dwayne Mitchell and Giselle Scholler are the 2 chairs. It's a quite complicated study, and it's really for patients with relapsed neuroblastoma and also with a brain tumor called DIPG, where you're using adoptive cell therapy, so you're using dendritic stem cells and also manipulating the T cells for patients to receive this therapy. It's a bit... has a number of moving parts. Patients get biopsied, the tumor gets sequenced, and then they receive kind of standard therapy while the sequencing results are back. They go on then to receive a targeted agent based on expression, while at the same time the cells are manipulated and they're eligible to receive dendritic cell vaccines. And then BCC 22 and 21, which we're very excited about for our patients with neuroblastoma. And I'll talk briefly about those. And so BCC 022 combines naxitamab with a drug called tipifarnib. And this was based on a lot of work with Dr. H.C. Wong's lab at Penn State. So tumor cells will secrete these extracellular vesicles, and these vesicles are really crucial, involved in cell progression, resistance to chemotherapy, and have a number of different actions. And he found that tipifarnib, which is an oral drug that's available, can actually suppress the secretion of those extracellular vesicles. And this is work from his lab. I don't know if the pointer can kind of work there, and it really shows that these are in mice models that the combination really suppressed the growth of the tumor cell, and then the mice that received the combination had an extended lifespan as well. And kind of the mechanism of action is that the combination of tipifarnib will help the immunotherapy work better. So we're excited, really excited. This study is now open and sites are enrolling. This is kind of the schema. It's going to be 6 cycles with tipifarnib and also with naxitamab given at these doses. That's the study design. And then Dr. Bahura is doing a study looking at a casein kinase inhibitor, which I'm going to kill the name, simetasertib, in combination with irinotecan and temozolomide. So we'll just say casein kinase 2, or CK2, is a protein that's overexpressed in a number of different cells, in leukemias, in solid tumors, and neuroblastoma as well. And it's involved in many of the pathways that are involved in cell growth. So she saw in her lab and also published literature that overexpression of the gene correlates with poor survival in both neuroblastoma and in Ewing sarcoma. And then she's done some preclinical work looking at, as a single agent alone, and also in combination, it really helps decrease the growth of tumor cells. And currently there's an open study in children with medulloblastoma. It's been well tolerated and has a good safety profile. So this study is going to be done in 2 steps. There's a phase 1 study that's looking at like a dose finding study of the best dose in combination with irinotecan and temozolomide. In sarcoma patients, vincristine will be added on. Once that recommended dose is determined, then it'll be opened up to a phase 2 study, both for patients with neuroblastoma and also with Ewing sarcoma as well. And talk a little bit about our biology studies within BCC. We have a biology study that's called BCC Bio 001. And the goal is to really look at sequencing of these tumors and also be able to collaborate and have tissues available for researchers around the world to be able to use. We have molecular tumor boards every week, and so patients are sequenced at diagnosis and also at relapse. We partner with Caris Life Sciences to do the sequencing. And our tumor boards are made up of pediatric oncologists, a pharmacist, a bioinformatician, basic scientist. And we look at the treatment history the patients received. We take into account what preferences does the family want oral chemotherapy, inpatient, outpatient. And then we look at any DNA mutations, but also RNA expression, and come up with a treatment plan for these patients. We look at pathways as well. And then in addition, from the biopsy, we also, it's a clinical component of the study, but also research in order to look at serum circulating tumor DNA and make this available to other researchers. We've sequenced close to 1,000 tumors, and last year had 131 tumor boards, and about 93 of patients had recommended therapy available through the molecular tumor board. This is just for 2024. We had like over 1,900 samples. These are available to researchers really around the country for if they have an interest for what they want to do. And then this really shows the majority of patients that are being sequenced, and the majority of patients that were profiled were patients with a diagnosis of neuroblastoma, but it's also open for other solid tumors as well. These are the cell lines and PDX models that have been developed that are available to research as well. So we really want to be able to collaborate and work together so we can make a difference for all our patients. Really want to, these are kind of our BCC members. We most importantly thank the patients and their families who participate in the research and our foundation support. And thank you so much.
anyone have any questions? No? Okay. All
All
right.
right.
Thank you,
Thanks.
Dr. Kraveka. Appreciate it. Thank you.